All right, well, welcome to another of the videos that we post quarterly to help support your teaching of your students in BioTechBuilder. I just finished speaking with Hiroko in the lab, and she and I agreed that we thought this one that could be most helpful and most relevant for right now would be a video that we made together that is part of our new professional development series on the DNA Foundations material. This video covers the math that is associated with our PCR experiment. So I printed out this lab activity from our PCR experiment that's part of the Foundations module that's part of DNA analysis, and it has a chart on the back which includes some of the calculations that your students should do in order to set up their polymerase chain reactions. I like this video for a lot of reasons, but in particular because it covers laboratory math like C1V1 equals C2V2. It gives you a chance to walk through the reaction setups in this protocol. It also talks a little bit about positive and negative controls in PCR and why we run them and what they can mean, why the results of those controls matter. And then finally, as I mentioned at the top, it is also included in our professional development training materials. This video is part of that for teacher training in advance, if you would like, of teaching this module to your students. So for all those reasons, I will show you that video next and post this material so that you can download it, this protocol, and as always, are here to answer questions that you have. All right, we are here to walk through the calculations that are associated with PCR. So we're going to start with what is on the second page of the protocol, and we've written up here on the board.
Just to be clear, for the teacher PD, you are not setting up the PCR, but the students, you will be with your students, and so we wanted to go through the calculations. So the first one on the protocol that's listed is a PCR reaction setup for the mini prep DNA. And there's a reaction table of all the components that will go in the tube, and there is one calculation here that they need to complete, which is to figure out how much of the 2x PCR master mix you need to add, and then secondly figure out how much water to add so that the total volume ends up being 50 microliters. So the students have completed or heard about making diluting solutions, and so here what you would use is a C1V1 equals C2V2. A very handy equation. And so C1 is the concentration of the stock, V1 is the volume of the stock, C2 and V2 are the final concentration in the volume. In the protocol, it's clearly written what the concentration of the final volume is, or final solution is, which is 1x, and the volume of the final is 50 microliters, and the concentration of the stock is 2x. And so what is left is solving for V1. And V1, if you calculate after unit cancellation, what you would end up is 25 microliters. And so then what they would do is to add 25 into this blank spot on the reaction table, and then subtract everything from 50. So 50 minus 25 minus 5, you get the idea. So what you get is 50 minus 30 minus 4 would be 16. Is that correct? Yeah. It would be 16 microliters of water that would add up to 50.
Yes.
And the students can add the math to check that their total is correct.
Yes. So this is the calculation that gets done for setting up the PCR with the mini prep DNA that is part of the teaching that you will do with your students. And again, as part of the PD, we are not setting up these reactions, but in addition to setting up the reaction tube, the experimental tube, which this we just described, there are 2 controls that the students will also set up as part of this PCR. And so the first control is a positive control, and that will be to verify that the PCR can work with DNA that is known to work in this reaction set. So this is with some positive control DNA that we know will amplify under these conditions, and that comes with the materials that come with this unit. And so in that reaction, which is the positive control reaction tube, there are many components that are the same volumes. We will again use 2 microliters of the forward primer, 2 microliters of the reverse primer, but the volume of that positive control DNA is smaller. We are only going to be using 2 microliters, is that right?
Yes.
2 microliters of that positive control DNA. The total amount, the total volume for this reaction is the same as the experimental reaction, so it still needs to add up to 50 microliters. These volumes are given on the protocol that the students will receive, but these are blanks that the students will have to fill in again. In this case, because the final volume is 50 microliters and the starting concentration and the final concentration of the PCR master mix is the same, we will use the same volume, 25 microliters of the PCR master mix, to set up this reaction. The thing that will vary is the volume of the water, which in this case, if I'm doing my math right, is going to be 19 microliters. So the difference, to bring it up to 50 microliters, that difference is made up with water, okay? One more quick version of this to just bring it home, the negative control for PCR. This is to show you that there is nothing in your reactions that will automatically give rise to a product. The negative control also has the same 2 microliters of each primer, but it has no DNA at all in it, right? So that should not, that should lead to no amplified product. The final volume is again 50 microliters. So once again, to go from 2x to 1x PCR master mix and to have a final 1x concentration, you are once again at 25 microliters of the 2x mix, but that leaves you now here with, what is that? 21, yes, 21 microliters of water to bring the final volume up to 50. All right, so that is the math that goes into these tubes to go into these reactions. What we're going to do next is show you the best practices and some pro tips for setting up these tubes, which have very small volumes in them, and some good ways to guide your students so that their reactions are more likely to be successful. Good? Good? Okay.